That was a very generous introduction.
I thought it was going to be a monologue.
Hello? Okay, I'm Charles Carmona, 1136, and I'm going to be Are we good? Say it again. Okay, Charles Carmona, number 1136, and I will be the interviewer tonight of our special guest. Can we roll the little video to start things off? Thank you. That's the right. Is it coming up?
I don't think so.
It just went.
No, okay, all right.
The video.
Well, let's just start with your background, Aaron.
Sure. Well, how far back do you want to go?
What inspired you to get into the minerals business, and what brought you to tonight?
Yeah, so I've always been interested in chemistry and physics, but in school I was terrible at that. And it wasn't until I took a course from Bob Downs at the University of Arizona in mineralogy that really inspired me to apply mineral physics, mineral chemistry, to really to find out ways of using that for environmental uses and pharmacological uses. And so I spent the rest of my career like finding ways of using minerals to address those kinds of problems. And that's where I really find my passion. So I go all over the world trying to find new minerals to do different things, or minerals in the museum. The museum is great because we have a huge repository, basically a library of minerals from all over the world. And it's just right there at our fingertips. So it's a fantastic place.
And what is your connection with JPL now, and what are you doing with that?
Yeah, so with JPL, that's interesting because we started off working on origins of life and trying to figure out when life started on Earth and what the mineralogical record tells us about that. But as we developed that, we started moving towards, like, how do we find life elsewhere in our solar system? Like, what kinds of things can we do to find life on Mars or the moons of moons of Saturn and Jupiter? And what does the mineralogical record tell us? Because if we were looking for life someplace else, maybe it died, maybe it's extinct. But minerals are persistent. Minerals stick around for millions of years, for the entire length of our solar system so far. And if we're going to find life, that would be great. But if we don't find life, our biological interaction, the biological fingerprint of what life was doing on a planet, is the only record that we have. And so that's a really key component. So we're doing origins of life work, but we're also looking for like biosignature detection on other planets and moons. So that's what our work is like at JPL. In order to do that, we have to go to places that are somewhat similar to other places in our solar system. And that's why we went to the Boulby Mine.
Okay, but before we go to the Boulby Mine, which is about the program, you also at JPL, you're on the program about habitability, and can you talk about that?
Yeah. Habitability was NASA's big push for a long time. They wanted to find places in our solar system that have habitable environments. So Earth, clearly habitable environment. That's data point number one. Data point number two is harder. We have to go to those other planets and moons to measure the mineralogy, to measure the geochemistry, to measure the fluids if they're present, to figure out, could they harbor life? Are they the right temperature? Are they the right pressure? Are there nutrients available? Are there places for them to hide during high solar radiation or high planetary radiation environments? And so that was NASA's big push for a long time. And that's what Mars rover is doing up right now, is doing a lot of habitability measurements. But I think we've now established that Mars is habitable. Mars had all the right conditions in its history and still has the right conditions to harbor life. now is the time to say, okay, when we know Mars is habitable, let's turn the page and say, is there life still on Mars? And if there's not, why not? And so that's what we're doing now at JPL.
Could we get that 3-minute video? Is that queued up?
I don't think it is.
All right.
I'll just go into it.
Okay. So the talk tonight is about the work you're doing a mile down.
Yeah.
In the Boulby Mine in. Tell us about it.
The Boulby Mine, I believe, is the world's deepest salt mine. It is a mine that doesn't actually mine salt. It mines polyhalite, which is used for fertilizer in the ground. So you just dig up this rock. It has all the right ingredients for fertilizer, and you just deposit it on the farmland. And that's what they do. And they have to dig deep in order to get that. The Boulby Mine is situated in a place, so the sediments are about 250 million years old. So 250 million years ago, what was northern England was an ocean. It was called the Celestian Sea. And that extended to what we know as England all the way to Poland. And that whole place was completely submerged. It eventually, because of plate tectonics, the plates moved around and the sea eventually dried up. But as it was drying up, it was depositing massive sequences of salt, which we call halite, gypsum, polyhalite, which is used as fertilizer, and all kinds of weird minerals. The interesting part about Boulby is that it's off the coast of England. You have to go right on the coast. There's a mine shaft there, and you go straight down, and then you start driving underneath the North Sea. But it's interesting because there hasn't been that much plate tectonics in that area for hundreds of millions of years. So it's relatively undisturbed. It's kind of this geologically pristine environment for 200 million years. And that's where we want to go. If there was life at the bottom of the Celestian Sea, and then it got just buried and buried and buried by sediments, could there still be life down there? And that's why we went down there to look for.
And how deep did you go? And
yeah, the mine shaft is a mile down. So that's us. That's the JPL team. I'm there with a goofy smile on. And then on the next slide, on the next image, this is the trip down into the mine with the rest of the scientific crew. So we have to get geared up. We have to get fit with oxygen masks. We have to get fit with emergency kits in case we get lost and run out of oxygen. So you'll see us wearing metal canisters on our belts. That's emergency respiration to convert CO2 into oxygen. And then we get into an elevator with a bunch of miners. And this elevator is huge. We all pile into it. And there's a check-in, check-out process. So you get two tokens when you first enter the mine. You get one token, you hand the first token, you get two tokens. You hand the first token to the elevator operator. So that way he knows who's in the mine. And then when you come out of the mine, you give the second token to him. So if there's two tokens on the board, then he knows everybody's out. So it takes about 7 minutes in an elevator that's going pretty quick to get down to about 4,000 feet below the surface of the Earth. And then we start hiking. So that's us hiking right there. And this is on Salt Road. You'll see machinery down there, like cars are passing by. So to get cars down there, they have to build the cars down in the underground mine. And we'll eventually get to the dark matter laboratory. And this is our home base. This is where we set up our shop. This is where we set up our microscopes. And this is the facility that we work out of. And it's a pretty, for all the mines that I've been in my entire life, this is the most posh environment I have ever been. The fact there's a bathroom down there was crazy.
so is it, you're down there all day?
Yeah, we're down there all day. The elevator only operates 3 times a day for the shifts of the miners. We go down there in the morning, and then we don't come back up until nighttime. So we're down there all day.
How many days did you spend on this latest trip?
A week and a half. So every day I was down there, and we take all our instruments down there. We take field instruments with us to various parts of the mine, and that's where we're doing a lot of the work. So the first few days was collecting. We just wanted to go out and collect as much as we could, because we don't know if something was to happen. So one thing that kind of made me aware that things can happen down there was that there was the emergency training miners. They go down there and they simulate mine rescues. So we have to go to these special rooms where if there is a mine fire, we have to go into these rooms and it seals us off from the mine fire. But then we have to learn how to breathe oxygen in that room. And I found out that there's oxygen tubing that comes from the surface into these special rooms, and there's not very many of these rooms that are there. So in some of the pictures that you'll see like this room with a bright green light. And I'm not sure why it's green, but green was very easy to see long distances down in the mine, so I think that's probably why. I imagine blue would be longer, but maybe there's reasons for it. So this is a picture of it. This is room number 17, and this is what you see. This is your safety. It's kind of scary looking. It's kind of alien looking, but this is the safest place in the entire mine to go to. And the slide on the right shows you what can happen in area, oh, the next slide, yeah, sorry. This area can show you what can happen. So this is a kind of a bad situation in a mine where you have fluid percolating through the rock. And where you have fluid percolating through the rock, that's where cracks happen, that's where faulting happens, that's where roofs can cave in. And so this is on top of an electrical generator, and all of those stalactites that you see are all made out of salt that are oozing in from the surface. So this is a problematic situation.
So it's percolating from above down.
That's right. And
is there pressure from below as well?
There is. We went to a couple places. So as we're looking for rocks that potentially harbor life, we have to be able to measure what current life looks like. Is there infiltration of fluid from the ocean above us? Is there infiltration of fluid from artesian wells and artesian pressure systems or hydraulic systems from below? So we went to all these different places where there's water just bubbling up from below us. We went to places where there's water flowing in from the top of us, make all those measurements so that whatever we do find, we know it's different than those other environments. And then luckily we had cars, because if we didn't have cars, it would have been a lot of walking, because once we go down the mine, we have to walk miles, or we would have to drive miles. It was like a 20-minute drive to some of these places at 40 miles an hour in a salt mine. And walking is just this maze of places.
So how long have they been digging this mine, and when did the scientists come and start the work that you're doing?
That's a great question. So the mine has been around for about 50 years, off and on operation, depending on the need for polyhalite for fertilizing. And right now it is in full swing. They are in the deepest part of the mine that they've ever been. It's over a mile down below. And they hadn't known that there's life down there until recently, until like the last probably 5 or 6 years or something like that. And so they're just digging through all these rocks, and they're excavating all the salt crystals that are really important for astrobiology applications, because they have bacteria still trapped in them. So you can imagine, like, what does this look like, right? What we're looking for rock. We're looking for a crystal that has pockets of fluid still trapped in it. And those pockets of fluid, that's something similar to what you're seeing on the screen, can harbor bacteria. But we don't know how long bacteria can live in these kinds of crystals. There's a mine in Mexico, the Naica mine, that has crystals that are 50 to 60,000 years old, gypsum crystals. And there is viable bacteria still trapped inside of those crystals. And so we know life can live for crazy amounts of time under extremely harsh conditions. And these beasties, what we're finding, we didn't even know what was alive. So we're just hammering away at rocks and trying to bring stuff to the lab. We're monitoring fluids down there. And we just didn't know what to expect. We're just looking at the crystals, and hopefully we find something. And as we move forward through the different rock units, we're starting to find interesting things. We're starting to find fluids. We're starting to find things that are moving within the crystals themselves, which was very exciting.
So that's like liquid-filled chambers with little bacteria that are floating in there.
Right.
Just a few a millimeter or a few millimeters, or
Much smaller than that.
even smaller
Yeah,
than
they're
that?
only 1 or 2 micrometers in size, so like 100th of the width of a human hair.
And you're able to see that with the laboratory equipment that you've set up?
That's right. So we built specific microscopes to do this. We built a holographic microscope so that we can take down there and measure fluid, anything that's moving in a fluid in real time. So with a visible light microscope, you have to move the objectives up and down to get things in focus. Well, you might miss what you are interested in. If there's only like 2 or 3 cells moving, you could easily miss that. But with a holographic microscope, you get the full volume of fluid all in focus, all at the same time. And then if there's something moving, then you can digitally go back and track it and trace where it's going and its origins. So that's what we were looking for, and it was amazing. So we found all these different places and all these different sites, and we did find life down there, which was kind of amazing. So I don't know if you can pass by a few more slides. So this is the digital holographic microscope. This is the system that was built in collaboration with JPL and Caltech. And you just pass a solution through it. And on the left is, what you're seeing right now is the computer screen showing the digital holographic image. And each one of those circles that you can barely see is a life form. That's still trapped that we extracted from the fluid, but we also can still see them in the crystal.
And could you estimate the time period in which it took?
Yes, we can estimate it, but we're still working on it. There's a lot of assumptions that have to be made when you're talking about life from that long time period ago. And you have to have a mountain of evidence to say that crystal formed 250 million years ago, and it hasn't changed since. And all the bacteria that's still in it is still 250 million years old. There's a amount of evidence you have to use to prove that. But what I think we have established is that life can persist a long ways away from solar radiation. It's not using sunlight to persist. There's little ecosystems of life down there. And how old it is, we don't exactly know. We're guessing it's around 100 million years old, but that we have to do a lot of work to still get to that point. We are paying attention to infiltration of water from the surface and infiltration of water from the subsurface. But a lot of the stuff hasn't been disturbed for a long period of time. Yeah.
So are these like unicellular little?
Yeah. They're just like little, here's a video of it, and if you see something streaking across the screen, actually in the top right corner, you'll see something moving around, and then it disappears. And at the bottom, you might see something like streak across. They're super fast little beasties. And that's cell motility. They're still alive, and they're still swimming around. Now, I know it's crazy, and they're also even more surprising, maybe in a couple more slides, you might see it. They're photosynthetic. Which blew our mind, because they've been down there for 100 million years or so. How do they still have the ability to convert sunlight into energy? Like, why does that even happen? And we still don't know, but they have the genes still preserved in their genetic code, in their DNA, to be able to convert regular light into energy. And we saw that happening in real time. So we take these white salts out, these bright white salts, and then just right out of the ground, haven't been exposed for 100 million years, haven't been seen in 100 million years. We take them back to the lab, and we can watch it turn pink, just sitting there. And so it's these bugs that are converting, that are trying to protect themselves. That's what they're doing. This pink color is called beta-carotene, and they're using it, they're secreting it out as kind of like a sunscreen. They're doing that to help preserve their biology, help preserve their DNA. And so when they see light, they're just like, oh my gosh, it's super bright out. I gotta put some sunscreen on, and that's what they're doing. And it's crazy. They do it in real time. Yeah. Oh, this is a picture of it. This is one of the test tubes that we have of fluid that was that was white at one point in time, and over the course of a couple hours, it turned pink. So we know there's living bacteria in there.
So what you are discovering a mile down in the ancient seabed, how can that relate to the mission of finding life on other planets and moons?
That's a great question. So the answer to it is we need to know what life is capable of. We need to know what kinds of extreme environments we find life and understand how it can persist. It's a mile below the surface of the Earth. How do they live? What energy sources do they use? Do they use radiation? Do they use chemosynthesis? Like what's driving their metabolism? And if we can understand that, it's like what drives their metabolism, then we can find other places in our solar system that don't have any sunlight, say like a moon of Jupiter or Saturn that has tiny, tiny amounts of sunlight, not really enough to drive metabolic processes. Well, since we know life on Earth exists without sunlight and can persist, what on those particular moons of Jupiter and Saturn could they use? They could use the radiation belts around the planets to generate food, nutrients to drive their metabolism. And that's all happening within the minerals. So that's the crazy part, is that there's this really interesting mineral biology interaction that we're discovering a mile down below the surface of the Earth that could easily be applied to other places in our solar system. This is a salt deposit, various types of salts, and we think there are salt deposits elsewhere in our solar system. So that's why Boulby was chosen.
So the minerals somehow feed the creatures.
It's potential. They might be potentially getting nutrients from the crystals themselves, although I'm kind of doubtful of that. We still don't have enough evidence. What we think is probably happening is that the crystals are protecting them from oxygen, and the crystals are protecting them from radiation so that their DNA and RNA can be preserved for really long periods of time. Oxygen is the worst thing that can happen to DNA. It oxidizes the DNA strands, and then it just breaks apart the cells, and it's really quite awful. So you have to have RNA present to go back and fix the DNA. And so if you have a lot of RNA there, then the DNA can keep getting repaired over and over and over again. And it turns out these bacteria have huge amounts of RNA present.
Are there other localities where this work is being done?
Yeah, California. We're doing this in California, the Salton Sea, and also
Underground?
Underground and Searles Lake. We just got back from a drilling job in Searles Lake. We drilled down to the bottom of the lake because the bottom of the lake is the sediments are supposed to be about 10 million years old. And every step of the way, we're getting sequences of rock. And so we're looking at how deep life went, what's the conditions like down there, and what was the environment like? Is there any kind of communication between bacteria, between one level and another level, that might cause mutations or something like that? So it's a great place to look for other types of life. And we're making all kinds of new discoveries of life forms.
Now, you said at the Boulby Mine, you're dating it back 250 million years?
That's when the Celestian Sea kind of fully dried up, was 250 million years ago.
Are there earlier is there older bacteria that have been found?
There is some evidence to suggest that some bacteria can persist, although not viable bacteria, for about 400 million years. And that is controversial. There is some evidence that supports it. There's a lot of evidence that does not support it. But that is really kind of pushing what we think is the maximum limit of how long a particular creature or an ecosystem can survive. 200 million to 250 million sounds palatable compared to 400 million, but it's still just huge amounts of time that these things can live. And they just live in these salt crystals waiting for the opportunity to be liberated, for the sea to come back, to dissolve the crystals so that the bacteria can then go out and do what bacteria do the best. And so we're collecting, we just found this spring, this is an example of one of the freshwater, saltwater springs that just came up. And that's the worst thing that miners want to see, is fluid flowing in a mine. But this is a rare opportunity for us to find some of these brine pools that are accidentally discovered by the miners. And so when this happens, the mining stops, scientists go down, we analyze the fluids, and then they can go back to what they're doing. Yeah.
What about other locations? Anytime there was an ancient sea, is that where it's likely that you'll, current, other than with the plate tectonics and seas opening up and
Yeah, that's another thing. That's a great question because but Boulby is pretty tectonically inactive, which made it great for looking for really old life forms. But I want to go to the other side of the sea, the ancient sea. I want to go to Poland and look at because Poland was very tectonically active. I want to see what kinds of differences we see in the mineralogy and the biology there compared to the other side of the ocean 200 million years ago. How much does tectonics influence changing of life over time? And so I'd love to go to Poland. I'd love to go into deep mines there and look for salt crystals, and specifically looking for these salt-loving bacteria. But really, any place that has evaporitic ponds, evaporitic salts, you're going to find me there at some point.
And maybe we'll find it on Mars?
Yeah, so that's the idea. Mars has a huge ultraviolet radiation flux right now because it doesn't have an atmosphere, or it doesn't have much of an atmosphere. So the ultraviolet radiation coming from the sun is not being filtered out by a nice ozone layer that we have on Earth. So the surface of Mars is just pounded by UV radiation. We don't expect anything to be living on the surface, so we're trying to develop a mission where we drill into the Martian subsurface. So with Boulby, we went down a mile. I don't think we have to go down a mile on Mars. That would be fairly impractical. But having ability to drill down 200 or 100 feet or so, 50 feet to 100 feet, would be amazing because you're getting away from the ultraviolet radiation. You're getting into the subsurface where chemistry and biology can still happen as evidence on Earth. So there is salt deposits there. There are ancient deltaic environments, old deltas and river flows, and those would be all prime places to look for beasties.
What's the we've had speakers at the club here talking about the future of space exploration.
Yeah.
But I guess specifically with JPL, do you have a timeline that you are following? Or people, maybe not you, but maybe your successors will be on some of the early Mars missions?
Human missions are super expensive, and a lot of robot missions they're not great. They're limited in what a robot is limited to what it can do. It's limited to what it's programmed to do. It would be great to have a scientist on the surface of Mars. That's how great science is done on Earth, is by people going out, picking up a rock, and making the connections between the environment of formation, the crystal, and all this other stuff that is just really, really complicated to make connections with. And computers so far haven't been able to really make those leaps of faith and judgment. I think this is a great crystal. Let me look at that a little bit more. And maybe artificial intelligence can do something like that in the future. I don't know. But that's what it's going to take, I think, in order for exploration to really be successful on moons in our solar system or on Mars. You're going to have boots on the ground. Whether that's my boots, I doubt it. But somebody's boots are going to have to be there.
Well, it seems to me that I read just recently that they expect to have humans on Mars by 2030. Is that?
If you talk to Elon, then yeah.
Gee, I gotta find his phone number.
I severely doubt it. I mean, the trip to Mars is brutal. It's 8 months, 9 months long. Yeah. You're subjected to all kinds of radiation. Zero gravity environments does tremendously bad things to the human body.
All right.
Yeah.
I accept that. We've talked about where we might find this on the moons of Jupiter, did you say?
Yeah.
Other similar deposits?
Yeah, that's another interesting place because the icy moons of Jupiter and Saturn are very different environment. They have fluid. They have liquid geyser eruptions that are happening right now kind of all the time. So we know that the interior of the planet or interior of the moon is liquid. But is there enough nutrients there to support? Life? So there is a mission right now I think it's called the Europa Clipper, that is going to go around Europa and start making those measurements, but it won't get there for a few more years. And so that'll be really nice to be able to ground truth. Like, should we even explore? Is Europa even habitable for life? What kinds of minerals are there? And I think those are going to be a lot of key questions that'll be answered in the future.
Have I missed anything?
I don't know. I got a couple more slides. We can flip through them.
Let's see.
Yeah, there's a lot of minerals that we find. There are a lot of weird minerals that we find down there. This is a mineral called boracite. The miners hate this mineral. I love it because this is a really interesting mineral that you don't find in too many places. It's not great for astrobiology, but for a mineral museum, for somebody who curates a collection, this is a really nice addition because it's kind of rare. The miners hate it because it's a hard mineral. So when they're drilling, they have these big like circular discs that they're pushing, like making tunnels in the subsurface. When they hit the boracite, sparks just go flying everywhere, and it really chews up the teeth on them. So they hate this stuff. On the next image that you see is what they're making their money on. This is sylvite. This is a part of what they're mining for the potash. So they're mining sylvite. They're mining polyhalite. You'll see that on the next slide, and these are the kinds of things that we were bringing back to the museum so that we have a record of what the mineralogical diversity is of this particular mine. This is polyhalite here. It's a very hard rock, and basically it's used as a fertilizer. You just grind it up and put it on the farmland, and that's it. So it's mine to farm marketing.
How many different institutions are in this program?
There's the Natural History Museum of Los Angeles. There's Caltech JPL. There is Edinburgh University is part of it, so I'm working with astrobiologists there at Edinburgh. And we also had a rover development team from UAE, United Arab Emirates, that went down in the mine, constructed a rover underground, and then started driving it around and making maps of the mine, making LiDAR maps of the mine, which was really interesting. And the next time we go down, we're also going to do some subsurface drone flying. And that's going to be a little bit more of a challenge. We're doing that for the miners themselves because we want to take the drones into some of these caverns that are being carved out by robots. And we want to send the drones in there essentially as a canary. We want to see what kind of gases are there before we start sending people into those areas. So that's where the drones will come in, doing sulfur detection, hydrogen detection. Yeah, and so this is a stalactite crystal. I wanted to have one because you don't see salt stalactites very often, and this is an example of one here. And there's also some rare minerals. This is the next slide.
But it's pointing in the wrong direction for stalactite.
It's hard to display it otherwise.
You could have flipped the slide.
That's true. I didn't do that, though.
For those who don't know the distinction, stalactite from the ceiling, stalagmite from the ground.
Yeah, it has to hold tight to the ceiling, stalactite, and mighty stalagmites are on the bottom.
Yeah. Okay.
Yeah.
Go ahead.
This is trombathite. This is a cubic salt crystal. It's a nice black crystal. It's also occurring in the potash deposit there. And then on the next one, it was a really interesting one. This is a volkovskite, and this is a rare formation, a rare mineral. It only occurs in a couple places in the world. It occurs in abundance at Boulby. And before I came to Boulby, I was doing my work, and I found a bunch of this on sale at eBay. So the miners are taking this out of the ground and selling them on eBay
to collectors,
whoever
mineral specimens.
They got this side hustle of selling crystals online. And I told the mine director this, and he's like, "That's great." I was like, "All right, that's cool." But it happened to be in the place in the mine that's been completely closed off. So this is an abandoned conveyor belt where they take some of the salt out of this, and this place is shut off from air conditioning. So how we breathe down there, for example, they have to pump fresh air in, and the pressure down there is about the same pressure as the surface of the Earth. So you're not getting, when you come back up from a mile, you don't get the bends and other issues that divers might get. But it was boiling hot down there. We couldn't spend too much time down there, so we just tell the miners, like, hey, we just need to collect some stuff and get out of here because it's like 120 degrees in pitch black darkness. So he's like, okay, so he gets this big stick and he starts jabbing at the ceiling of the mine, and I'm petrified. I'm like, and it's caving in. And he's like, okay, here you go. I'm like, oh, please don't do that again. We don't need, this mine is not, this part of the mine is not monitored anymore. It's now completely flooded by the ocean. They let the ocean into this part of the mine. And it was absolutely terrifying. And so on the next slide, you'll see us driving back from this area.
Real fast.
Real fast. No, he actually slowed down so I could take a video. We're going about 40 kilometers an hour, 50 kilometers an hour. And what you see on the left and right side is the support beams. So normally in a salt mine, the salt is quite strong. You don't need supports. But over time, the mine will start shifting and things will start moving around. And so they put in these wooden beams. That's the first defense. The second defense is that you see is the steel beams on the ceiling. And that is the last line of defense. They only have 2 lines of defense, wood and then steel beams on the surface. And when you see the steel beams on the surface, you know you're in a bad area, and you know you have to really pay attention to the ceiling indicators. So you might see like these little white things on the ceiling. Those are measurement devices that show you in millimeters how much the ceiling has moved from day to day. And you really have to pay attention to it. Yeah, it's scary. We were in a part of the mine.
So the economics of the mine they have huge pieces of equipment that are scooping up cubic meters at a time,
Yeah.
and then there's conveyor belts to take it to the surface.
Yeah, that was the original part of the mining process and you can see on this image is that these are like the drilling mechanisms that they have. And that was when they were in the sylvite part, which was a little bit softer. Now they're in the polyhalite part, which is like hard as granite. It is hard material. So what they do is they bore 2 holes next to each other, parallel. And what that does is it forms this hourglass or this double concave thing right in between the 2 boreholes. And then they just wait. Everybody comes back out and they wait. And what happens is the pressure from the ceiling crushes that part, and it just turns it into rubble. And it's a very explosive process. Very dangerous kind of mining, but that's what they're doing. They're just drilling these holes, wait for the ceiling to collapse, and then pull out the material from that.
There's no explosives that they use.
No explosives. Yeah, they use the pressure of earth above as their crushing medium. And how they pull it out is that they use these big diggers to get the material out of the mine. And they have to construct all this stuff down in the mine because they only have the one elevator taking you up and down. And it's not a big elevator. You can't fit a car in it. So they have machine shops and they have automotive manufacturing down there. Yeah. It's a big process.
How many people are down there during a typical work day?
There's about 100 people down there. That includes the miners and any kind of research scientists. And then they cycle that out halfway through the day. So a shift typically is around 6 hours of hard mining, and then you come back out, and then you go to sleep, and then you come back. So a mine operates 24 hours a day with miners coming in and out of the ground. Yeah. And so just to give you a sense of how dark it is, that's 3 people standing next to each other. And it's difficult to get a picture down there because it is so pitch black. And I remember one of the miners, right before we took this picture, after we took this picture when I was very happy and I was a big smile on my face. And he's like, okay, everybody turn off their headlamps. We turned off, it was just 3 of us, and we turned off our headlamp. And you just listen to the earth move around you. So you can hear the mine shifting and twisting as you're in this area. And plus we're knee deep in water at the same time we're doing this. So he set up a really nice.
So is that phosphorescent?
Yeah, that's our high-vis jerseys that we had to wear.
So there's no light that's exciting that.
Oh, sorry, there was a flash on the camera. Yeah, that's just a reflection.
Flash.
Sorry,
Okay.
not phosphorescence. That's just a reflection. Yeah. Yeah. So that was the Boulby Mine. I don't know what the next slide looks like. That might be it.
Is that the end?
It could be.
Okay.
So then we just pack up and then we head back to the States, and we have tons and tons of salt crystals. Then go through, do the DNA sequencing.
When were you actually there?
I was there in 2020, right before the pandemic. So we left the mine as the world was shutting down. I didn't think I was going to get back into the States. And then we went back again in 2022. And I plan to go back again in 2024 or 2025.
Okay. So do we have some more slides, it looks like?
Yeah, this is us packing up in the dark matter lab, trying to get everything organized and just try to get everything shipped out in one piece. But that's it. So we just collect as much samples as we can. Yeah.
So shall we take some questions?
Yeah, we can take questions. Yeah, that's the end of the trip.
Please, we have a microphone.
So it's fascinating that the life forms can live for 100 million years or 200 million years. Are they anaerobic? I mean, they exist without any kind of oxygen?
That is correct. I'm told to talk to the camera. So the camera's right over there. Yeah, that's correct. They are anaerobic, but we don't know exactly how they metabolize nutrients. So if there's any, we do know that if there's any oxygen there, that they will get stressed and they might produce some other chemicals to try to get rid of the oxygen. So beta carotene is one example of that. It's an oxygen getter. Once it secretes oxygen, once it secretes the beta carotene, it just goes out and grabs as much oxygen as possible. And so that beastie can live in an anoxic environment. So that's one thing that we do know is what happens within the fluid inclusions themselves. Now, in the brine fluids that's flowing, we still don't know exactly how they live. So that's what we're still trying to figure out.
Has it been possible to remove the organisms from the salt and raise them in laboratory cultures?
That's a great question, George. So with the gypsum crystals from the Naika mine that are 50, 60,000 years old, yes. There are groups that have been successful in extracting out the archaea and bacteria from that. We have tried many times with these. We've either killed them every single time, or they are not viable COSI in the fluid inclusions. Now, with the fluids flowing around, yes, we have been able to feed them nutrients and to get them to propagate. So NRC1 is a type of bacteria, is very similar to these that we grow in the lab, and we feed these bacteria the same nutrients as we feed NRC1, and we are able to grow them. They mutate pretty quickly. And so it's quite challenging. So what we're also trying to do is we're also using nanopore technology to extract out the single cells of bacteria and sequence a single cell of bacteria for its DNA. That way we don't have any mutations. We don't have to worry about feeding it food. We also don't have to worry about the cells lysing. So I get questions all the time. Are we going to start like the next worst case pandemic on the planet by releasing these 200 million year old bacteria? Very, very unlikely. These creatures have to live in the most salty environment that you can possibly imagine. You can imagine an ocean being salty. That's nowhere near salty enough for these things to live. They have to have salt. And so as soon as we take them out, as soon as you crack the crystal and just regular air interacts with them, the cells explode on contact. They're just so hard to keep alive. So yeah, we're trying all kinds of techniques, and we think they're most closely similar to NRC1, but we're still working on it. There's so low biomass, it's quite challenging. Great question.
So I think that it sounds like you're not going to be giving information for Jurassic Park 6. Is it number 6 they're up to?
Well, it wouldn't be very exciting having these little invisible things floating around.
But who knows what they could turn into. Anyway, we have some more questions.
So it's called Dark Matter Lab. Did you find any? I assume you weren't looking, but why is it called that? And my real question is, if they're 100 million years old plus, I'm assuming they're reproducing. If they're reproducing, there's got to be evolution because there's got to be transcription errors. And are you looking at that? You have any thoughts?
Yes. It's called the Dark Matter Lab because that is the group of scientists that are down there. So they carved out this area so they can do neutrino detection and other things like that. So there's this whole lab down there dedicated to, this very clean room dedicated to those kinds of studies. And they have the dirty area, which we were allowed to be in, to do whatever sample prep. But we use the facilities of the dark matter lab, because dark matter research gets tons of funding from from those groups. Astrobiology doesn't get so much funding. But as far as like the DNA sequencing, transcription, mutations, and things like that, that's not my area of expertise. So I'm really interested in. So I don't know the answer to that question, but I'm really interested more in the mineral-microbe interaction. Like why do microbes want to go inside of crystals? Like what is that driving force? And why do they, so why do they do that? What is that interaction between them? So what is gas migration in and out of the crystal? How does the crystal filter out radiation? Those kinds of things. So that's a great question, but I don't really know the answer to it.
Yeah, I have a similar question about the biology. Are all prokaryotes? There's no eukaryotic cells that you found? Are there other? What's the range of life that you found there?
It's very low diversity. It's prokaryotic mostly, and
Can you define that?
no.
No nucleus.
Yeah, it has to do with no nucleus, like the cellular structure of it.
And do they have mitochondria?
Right.
Do they have mitochondria, and how do they compare with other life forms on Earth? Have you run the DNA sequencing to see level of relatedness?
Yeah.
How do they compare with other deep salt bacteria that you found in different parts of the Earth? Are they related?
They are very much related. There's not a whole lot of diversity when it comes to these halophiles. There's more halophilic diversity in the archaea than there is in bacteria. There are very few halophilic bacteria. And what we're finding here is bacteria, very little archaea. Which is similar to what we're finding in the deep deposits of the Searles Lake. And we're also finding a lot of bacteria in the Great Salt Lake as well. So there's a lot of similarities to this. And the interesting thing is that to me, what the most striking similarity is that they all seem to be photosynthetic. It doesn't matter how deep we went, they all still had the ability to photosynthesize. What does that mean? I really don't know. But it's an interesting clue of what might be taking place down there that we just haven't been able to measure yet.
Okay, it's another biology question.
Shoot. I'm a mineralogist.
I know. Now, you've mentioned DNA sequencing a couple times, and I was just curious if you could maybe elucidate a little bit more. How big is it? I assume it's not very big. It's a unicellular organism. Do you know the size of the genome or anything
I don't know.
interesting about it?
Yeah, that's really interesting. I don't know the size of the genome. In fact, we're just now getting that data back. And so hopefully we'll be able to answer those questions really soon. But it's still an unknown. Because there's so little biomass, the biologists are trying to figure out ways of concentrating the biomass into more than just one cell. And that tends to be a little bit trickier. So we're doing this with the eye to how do we explore life on other planets? What is the lowest amount of biomass that we can have to really do some sequencing or 16S, some kind of like gene typing? So those are things that are happening in parallel to what we're doing. So this is a good example of like what is the minimum amount of biomass that you need? It's more than one cell. I can confidently say that. But what 10 cells, 100 cells? Like what do you need? I don't really know.
Can you tell us a little bit about? So these are 250 million-year-old bacteria.
Ish, yeah.
And they developed a relationship with some mineral crystals.
Yeah.
What was the world like 150 million years ago? Can you describe the environment and how they then got sucked down this deep in the earth?
Yeah, they were once at the surface of the Earth. So 250 million years ago, this was a sea. It was pretty hot 250 million years ago. There probably wasn't any ice caps on the Earth. It was a major warming period on the planet. So we're finding, dinosaurs in the Arctic, for example, like that 200 million years ago. That's the kind of, like reptiles in the Arctic and Antarctic. That's the kind of time frame we're talking, like kind of like what you can visualize. Planet is very hot. And because there's no ice, there's a lot of water. And there's a lot of these seas and these bays and all kinds of intermittent, like these transient sort of lake environments. And as the planet started to cool off, that water froze. And the seas are starting to dry up. And so as the seas are drying up, and there's sequences, it's not just a one-time event, there's probably multiple warmings and cooling periods. And so as the seas are drying up, then there's lots and lots of sediment, these salts that are precipitating out of these oceans. And as the salt precipitates, it encapsulates the bugs. But what's interesting is that as you cycle an ocean multiple times, you go rehydrate, you put water in it, you take water out, you put water in it, you take water out, the chemistry changes. And you start precipitating out different kinds of minerals because of these different kinds of sequences of mineralization. So we're studying that in the lab, like how can you grow a crystal and get it wet and dissolve it, grow a crystal, dissolve it, grow a crystal and dissolve it, and how do the bacteria respond to that? And it's super interesting. The bacteria learn, or I think they learn based on video evidence, is that they just swim right up to the crystal. Like at first, when we're doing the rehydration and dehydration, the bacteria just kind of swimming around and not doing anything. But you do this a couple times, and as soon as the crystal starts growing, you see all this bacteria swim right to the crystal face and just stay there until the crystal just overwhelms them and just plows right over top of them and incorporates the bacteria inside the crystal. And we did this experiment a bunch of times, and every time we did it, we saw the similar thing. At first, bacteria just don't even care about the crystals. You do it 5 or 6 times, and the bacteria find their way to the crystal like right away. And it was bizarre.
So small time scales.
Yeah, they were talking tens of minutes we're doing this. I think the longest one I did was a couple hours. And it was...
And the little microbes are going like in between the crystal forms, so they're like trapped.
You can imagine like an aquarium. So on the right, so if you go to the next slide, this is a good example. So they're like crystal aquaria. And when you look at the little pockets of fluid that are trapped inside of them, you can still see the bacteria still swimming around. So like this one right here. The mouse is following a little beastie inside of the crystal. And this is only 50 microns. The mouse is about 1 micron in scale, for example. And so they just swim around. Could be Brownian motion, which is just random molecular motion, or it could be cell motility, which is cells actually driving their own motion. It's really hard to tell that in such a small, confined space because they're just bouncing around anyway. But yeah, so if you dissolve this crystal, then they're allowed to swim around. And then when the crystal grows again, they get stuck back into these fluids that are trapped inside the crystal. So it's very interesting. So every time we did that, they became... I don't think they learned. I think there's probably, that would be a lot to expect of a bacteria, a single-celled organism without a nucleus. But what we're thinking is that the fluid chemistry changes, that there is this chemical gradient between where the crystal is and the chemistry is actually in front of it. So there's actually some polymerization that's happening in front of the crystal, and the bacteria can sense it. They sense, oh, this stronger salt signal over here, so they start swimming towards wherever is the saltiest, and that's how they stay alive.
Okay, I got 2 quick questions. Have you ever actually seen them reproduce?
Yeah, for modern ones we have. For modern ones we can watch them reproduce cell division pretty quickly. And at first, it's interesting. At first, these bacteria don't look like round little blobs. They're very much rod-like shape. And once the water dehydrates, they immediately break into smaller cocci, smaller spherical shapes, and that's the bits and pieces that get entrapped inside of the crystals. And they will stay like that for a long time, and then when you rehydrate it, they still stay as cocci. I've actually never seen them go back to a rod-shaped form again, but they still do cell division.
Now, when they're trapped inside the crystal, kind of like a little terrarium of sorts, so they're eating other organisms that are in there. They're eating each other or something.
That could happen.
I mean, so the nutrients, there's a fixed amount of biological nutrient in each crystal.
Yeah.
And that doesn't change. So it must transform from one to each one, and then it dies, and then something else eats it, and it's the same stuff recycled over and over and over.
We thought the same thing too. Like, how do these things eat? And one thing I did measure, kind of by accident, was that I took the same crystal and I just like, well, what just happens over a course of a month or 2 months? I can't remember how long. It was several months long. And I looked at the same fluid inclusion in the crystal over time. And what I found was that the fluid inclusion shape changes. So as the bacteria are circulating nutrients and fluids, the crystal is starting to dissolve. So the crystal dissolves and the fluid migrates a little bit within the fluid inclusion. So the fluid inclusion doesn't stay in the same place all the time. It slowly migrates as you dissolve one side of the fluid inclusion and the other side recrystallizes. You dissolve, it recrystallizes. It kind of like marches along like that. And it takes months for that to happen. And I just discovered it by accident just because I wanted to see how one fluid inclusion was behaving over time. The fluids become more round over time, and it's really kind of an interesting, weird dynamic that's happening within the crystal that we don't quite understand. Oh.
Yeah, I've understood about 1/250 millionth of everything you've said. But I'm curious, what's the difference, if there is any? I've been to both. Is there any life in the Great Salt Lake and in the Dead Sea?
Yeah.
You call it the Dead Sea, but obviously it's not dead.
It's not dead.
If I've understood anything of what you've said.
Yeah, you're exactly right. It's not dead. In fact, in the Atacama Desert, we've gotten salt crystals from the tops of the mountains in the Atacama, the world's driest place. You flip a rock over, there's living bacteria. It is insane how these things can persist for so long under such extremely harsh conditions. We're just touching, it's just the tip of the iceberg of the knowledge that we're generating. Like, how does this microbiology really take place? And I really feel passionate that it's this mineral interface between the biology, like what's happening there. If you flip a rock over and there's biology like stuck to the rock, they're using the rock for some reason. What? Protection from radiation, food, we don't know. Yeah, it's crazy.
I'm going to take one more question over here.
I'm not going to ask you about critters.
Okay, good.
You've talked about several different types of salts. What does the chemistry of those salts say about what was happening in atmospheric chemistry or heat or whatever at the time it was deposited?
It's a great question. So there are various types of salts, and I've mostly been just talking about the halite salt, which is just our table salt. But if you go to the grocery store, there's Epsom salts, right? That's a salt as well. That's a magnesium sulfate salt, and it forms if a water was rich in magnesium and sulfur and produced these crystals. The problem is Epsom salts, even though it says Epsom salts on the outside of the bottle, it's not really Epsomite. Epsomite is, I'm just going to have to say it, it has 7 water molecules per formula unit. But I know I had to say it. But that's one mineral. But if you leave that sitting out, it dehydrates. It dehydrates to a mineral that has 6 water molecules per formula unit, which is hexahydrate. So when you go to the grocery store, you're buying hexahydrate. And if you add a little bit of water, it turns into Epsomite, which is pickling salt. So that's one type of salt, which we think might be present on the surface of Europa, really, really cold places. There's another one that's called mirabolite. This one hits a little bit closer to home because mirabolite forms at the bottom of like concrete. So if you have in a humid environment, you have a concrete sidewalk, you have sodium sulfate sitting underneath the concrete, and it's crystallizing mirabolite. That's bad for the concrete. It's getting the sulfur, it's getting the sodium as nutrients from the concrete, it's breaking it down. But mirabolite is not stable. It quickly converts. Like if you just took up that mirabolite crystal and you held it up in your hand, it would turn to a white powder immediately, from a crystal clear crystal to a white powder within minutes or even seconds. And that mineral is called thenardite. So it goes through this cycle, and the concrete industry really hates this stuff because this is what breaks down concrete over long periods of time. But this is a great place for bacteria to hang out in these sort of intermittent cycling events, which is really kind of interesting. So stagnant events, bacteria don't really like that much. They like more of these transient sort of areas, and so thenardite and mirabolite are great examples of this. That's another kind of salt, sodium sulfate salt. There's a whole bunch of these kinds of salts I won't bore you with. But but they're all different at different planetary conditions. May not necessarily be stable at Earth's surface, so that tells us what the environment is like. So if you find a place that has stable mirabolite, thenardite, you know what kinds of pressures and temperatures you're talking about. Does that answer your question? All right, mostly. Sorry. First mineralogy question.
Okay, I think we've come to the end of the evening. Rich, do you want to?
Just wrap it up and go play a video.
Okay. All right, well, thank you, Aaron. You have, I don't know if it's the very first time we've had this type of a discussion at the Adventurers Club, but it certainly is interesting, and thank you for coming, and you'll come up with another topic maybe next year?
I hope you invite me back.
You're on our list.
All right.
Thank you.
Thank you.
All right, stand here, Aaron, real quick. I got a certificate for you. Oh, my gosh.